I'll get started. Thanks for that introduction, Pam. I'm Jacqueline Robinson. I'm a genetics researcher at UCSF, and I work in Dara Torgerson's group. I mostly work on wildlife genetics, but we also work on a lot of human genetics projects there. But lately, we have had the joy of focusing a little bit of our research on the famous San Francisco red-masked parakeets. So I will be referring to this species as red-masked parakeets, but I know that they are also commonly called cherry-headed conures. Hopefully I won't get the terminology too mixed up, but I'm trying to be consistent. So it's a pleasure for me to be here joining this holiday event to give you an update on what we've been doing and finding with our genetics research. So just to kick things off a little bit here, this is a video that I took almost exactly a year ago. I just happened to be visiting a friend who lives next to Lafayette Park in San Francisco, and I heard this incredible cacophony coming from somewhere. And if you can hear that, that's a little bit of the sound from the video. But I walked over to this tree that was emanating all this noise, and it was full of parakeets. So the parrots have become actually quite famous, especially lately, now that they are the official San Francisco animal. So today I'm going to give a little bit of background about this population and tell you more about what we are hoping to learn from our genetics research and give you an overview of the phases of the project. The first phase is pretty much complete. It's the sequencing of the full genome of our representative red-masked parakeet named Acorn, and you'll hear a bit more about Acorn later on from Dara. And then I will cover some of our results pertaining to the population of the San Francisco flock, their genetics, and what we've learned about levels of inbreeding in the flock, relatedness amongst individuals, and even the hybridization, the sort of hybrid ancestry of the flock, which is not pure red-masked parakeet. And then I'll just wrap up with some conclusions, and hopefully we'll have some time for your questions at the end. And this picture is, I believe Dara took it. It's from one of the early morning releases where I believe that tree actually has a bunch of the flock. I think that's Sue Bierman Park in San Francisco. So the flock in San Francisco is not, as I said, pure red-masked parakeet. Just based on observation, we know that there are mostly red-masked parakeets, but there's also, there was at least one mitered parakeet in the flock that then reproduced, and now the flock is kind of a mixture of red-masked and mitered parakeet. And also there was at least one blue-crowned parakeet observed in the flock, but as far as we know, it hasn't made a genetic contribution. But this is some of the things that we're interested in learning, or sort of like answering these questions with actual genetic sequence data. So like many San Franciscans, the red-masked parakeets did not sort of originally come from here. They came from elsewhere, up until I think 1992, when the exotic bird trade was finally made illegal in the US. It was pretty common for wild birds to be trafficked from their native ranges and brought to other places like the US. So the parrots that we have here are the descendants of some, well, I don't know if lucky is the right word, but some escapees, either from homes or from pet stores. And one thing I wanted to point out here is that the three species that have been observed in the San Francisco flock, they originate from very distinct geographic areas in South America. So they're completely different, divergent species. In their native ranges, they would never come into contact with one another, but they've all ended up in San Francisco and joined, a little parrot melting pot here. So what are we actually interested in learning about the parakeets using genetics that we can't really learn about in other ways, or that would be very difficult to know? So on a basic level, we're interested in knowing about the patterns of genetic diversity in the San Francisco flock, how much diversity there is, how similar is the flock to other parrot species or other flocks in California. We can learn more about exactly how individuals in the flock are related to one another. And another thing that we can do is if we get um, if we get biological samples from individuals, we can tell if the sample sort of came from that individual. So this might be helpful if, for instance, a bird from the flock gets injured or is ill and then ends up being rescued, coming to the vet. If we already have that individual sampled in our database, we would know, oh, this is that individual. It's easier, I think, to use microchips, which is the technology that's being implemented now, but technically we can tell if samples came from the same individual or from related individuals. We can also use genetics to learn about the history and the origins of the flock, which are somewhat mysterious. We know that there have been wild parrots sighted in San Francisco going back to the early 20th century. The flock seems to have gained notice and really become established sometime in the late 1980s, by the early 1990s. But we don't really know the specifics. There are a lot of stories about how the flock may have gotten started, but it's sort of not really known for sure. Genetics could help us figure out with more specificity, how many generations back does the flock go? And specifically what species, the species of the individuals that have contributed to the individuals that are in the flock today. Another important question that we have is, given that the San Francisco flock is sort of small and isolated, are we seeing evidence of inbreeding in the flock? And I've touched on this already, but hybridization is something that we're interested in learning more about. And in the long run, we might actually be able to use genetic information to learn more about the health of these birds. So today I'm going to be mostly talking about genetic diversity, inbreeding, and hybridization. But we have a lot more analyses planned sort of in the long run. And I'm really excited about where this project could be going. So phase one of this project, where we really had to start from the ground up, was in getting, for the first time, a high quality, full, complete DNA sequence from one of the parakeets to use as the scaffold for all of our future genetic analyses as we sequence more individuals. So we were able to do that with Acorn's genome, and we'll talk more about that shortly. Phase one is more or less complete. Phase two of the project is what we've been working on for the past, I want to say, 10 months or so, where we've been sequencing multiple individuals from the flock, mostly living birds that are in foster care. And that gives us a better perspective on, the flock in general, not just one individual. And that's what I will be talking about later on in the presentation. Phase three this is more future work, further sequencing of more individuals to expand the data set. And one of the things I'm really excited about is incorporating individuals from beyond the San Francisco flock as well. So we can kind of put our population in the context of parrots, across California, for example. And the little icons here are representative of the kinds of biological samples that we are collecting in these phases. to obtain DNA. And in the early part of the project with phase 1 and phase 2, we need really high quality DNA in high quantity. So the best thing for that is to have tissue or blood or even molted feathers. So some of our work has been just experimenting with what kinds of samples can we get DNA out of. And I will probably reiterate this later, but it's important to note that we are not specifically going out and trying to collect samples for our project intentionally. All the samples that we've used are things that have been obtained opportunistically, like if there's leftover samples or molted feathers, things like that, or deceased individuals. In future, we're interested in perhaps getting permission to collect molted feathers from the ground around where the flocks roost, maybe even sample feces and kind of use that to do a much more comprehensive survey of the population. So I will start by talking about the first phase of the project, which, as Pam mentioned, we presented on earlier in the year, but I will recap that today. And phase 1 was, again, sequencing the full DNA, like full set of DNA from one individual to get this really high quality, complete sequence from Acorn specifically. Dara, are you on? Do you want to talk a little bit more about Acorn's background and story and how we came to do this?
Yeah, so Mickaboo often gets contacted by San Francisco Animal Care and Control. So oftentimes, while the flock is doing very well, every so often they, get into trouble. And often city workers or Good Samaritans, they find them. And, the real lucky ones get to Mickaboo in time, and we do our best to create a sort of birdie ambulance and take them to avian vet for the needed care, and hope that they will recover and be able to return to their homes. So actually, this was our office at UCSF is right near Bryant Street, San Francisco Animal Care and Control, and so I was just about ready to pack up, and then we got a call saying that there was a conure here. Here's a photo of Acorn in the box, and they weren't doing very well, and could we get them to medical care? And so I arrived at San Francisco Animal Care and Control, and picked up the box, and they had written "angry conure," and I thought, well, this is a good sign, because an angry bird is a bird that's doing okay. And so then got on the bridge traffic to try to get them to a vet, and a whole team of other volunteers was sort of behind the scenes figuring out, okay, well, where should they go, who's available, what vet can take them right away. And so loaded up in the car and brought them to the vet. I, of course, was sort of peeking in a little bit to see if they could see them, but we never open up the box until we really sort of get to the vet. Next slide, please. And so when Acorn came in, he was missing some feathers on the right side, open wound on the back of the tail, which might have suggested like a cat attack or a hawk, and also bruising on the beak, which often indicates an impact injury. But also very marked clinical signs of bromethalin poisoning. So this is a rat poison that unfortunately is in use in the city, and we do see a lot of birds that come in with this typical head tilt. So it causes neurological damage. And so in this case, the head tilt was past 90 degrees. And Mickaboo was part of a prior research study that actually identified and confirmed the finding of bromethalin in a lot of these birds that passed away. And so really there's no treatment beyond supportive care. And, while we sort of understand some of the clinical features, it's really sort of difficult to know, okay, are they going to improve or are they going to decline? And so typically this can take, 1 to 3 months of supportive care. And unfortunately, Acorn was one of the unlucky ones, and passed away a month later. So, despite the best care that could be provided, unfortunately, it was a little bit too much for Acorn.
Thanks, Dara. So although Acorn passed away, in some ways they live on through having their complete genome sequence that is facilitating all the rest of our genetic work. It's again, it's not necessary for us to have a deceased bird in order to do genome sequencing, but given that Acorn had passed, and we weren't really quite sure exactly how well the sequencing was going to go, it was helpful for the project to sort of have a whole bird that we could get samples from and start doing the genome sequencing and assembly. So here's some pictures just showing various stages of how this works. There's a picture in the upper left of what our lab looks like. That's where we take biological samples like blood or feathers or what have you, and then extract the DNA using, pipettes and various extraction liquids and things like that. The middle picture on the bottom there, if you can see in the tube, there's this little cloudy area. That's actually a very dense sort of clump of actual purified DNA from a sample. And we can then do further lab prep work with that in order to load it onto a sequencing machine that is then going to spit out all these chunks, small chunks of DNA sequence that we then have to kind of put together to create the genome. And all of that part is done just computationally. So in the bottom right is a picture of me looking at some actual DNA sequence data. Like, you can't really see what I'm looking at, but it's things like AATCGAAA, just a huge string of letters like that, the DNA bases. So a sort of visual way of showing this is what we get out of that sequencing machine is all these fragments of strings of DNA bases or letters. And the challenge is to then put that together into the actual sequence of this individual's DNA with all of their chromosomes. And an analogy that I think is helpful for for understanding what we're doing here is, imagine you wanted to read a book and a friend gave you a book, but that book had gone through a shredder for some reason. And you were then your task at that point is to try to recreate the book by finding, overlapping parts or parts of the book that go together. You're going to build that up into sentences, chapters, and then eventually have the whole book. That's sort of what we're doing with creating a genome for an organism for the very first time. So that was done successfully with the help of our colleagues at Stanford, Dr. Bernard Kim and Hannah Gellert. And that laid the foundation for our population level analysis of the wild flock with, I kind of think of it as a pilot phase of the project. This is where we're starting to learn things about the nature of the flock, how individuals are related to one another, and so on. And for that, we have used blood and molted feathers. So how exactly are we getting these samples? As I said, we are not allowed, or we don't have sort of regulatory permission to go out and specifically catch birds and get samples from them. So what we're doing is working with vets and foster carers with Mickaboo to get basically leftover samples that would otherwise be discarded anyway. So things like the vets are taking blood for their health, like taking care of the birds. If there's any leftover samples, we can use that. That's what's shown in the picture on the top right. We can also get tiny, tiny little blood spots if through the routine care of the birds that becomes available. So if any of you have ever trimmed birds' nails, sometimes you get the quick and there's a little bit of blood that comes out. We can, take where we've absorbed some of that with a little bit of paper and use that to extract DNA. And as I said, we found that we are also able to successfully use molted feathers. So we're not harming the birds, or they don't have to go through any additional sort of stress for us to get samples for this project. We're just taking advantage of things that are already available. So the genomes that we have sequenced to date include Acorn's genome, and then we have 21 additional samples from the San Francisco wild flock. And then just because they happen to be available, we also folded in a couple of samples from some Sunnyvale and Palo Alto birds, and also just a pet red masked parakeet that was not a part of the San Francisco flock at any point. I then also obtained some genomes from an online public genetic database from the red-masked, mitered, and blue-crowned parakeets to use as kind of reference points for our analysis. So we could, look at things like how similar are the San Francisco parrot genomes to these other independently sequenced birds in the public database. So one of the very first things that I look at in a project like this is how much genetic diversity is there in the genome sort of at large. So I'm not going to get into the nitty-gritty details of exactly what we're measuring here, but suffice to say, we can sort of march along the genome and quantify how much variability there is within and between individuals. So this is a picture of diversity within Acorn's genome. And the reason there is diversity within this one individual's genome is because birds like us have 2 pairs of each chromosome. One member of each pair comes from the mother and one comes from the father. And there's some minor differences between those chromosomes. And those are the genetic differences that we're quantifying here. So on the x-axis of this plot is just sort of the coordinates of the genome assembly, all the different fragments that we have. And we can analyze those patterns to learn about the background of these individuals and of the flock at large. So a very prominent pattern emerged once I started looking at more individuals, such as Julian here, who I think is in Dara's home right now. But Julian is one of the individuals who showed a very distinctive pattern of inbreeding. And how I could determine that was by seeing that there were very large regions of Julian's genome that had no diversity, which meant that Julian's parents, his mother and father, were closely related and had this exact same DNA segment from a recent ancestor that they both shared. So I saw this pattern in pretty much every individual from the San Francisco flock to varying degrees. To give a summary of what I found, here is a graphical representation of an example of inbreeding, where in this case you have 2 parents that had 2 offspring. Those offspring then mated with one another and produced the individual in orange at the bottom there. That individual is inbred because its parents are closely related. So we can use the genome sequences to measure the extent of that inbreeding and quantify how closely related the parents were of these individuals. And in the San Francisco wild flock, every single bird had a signature of some level of inbreeding, indicating that its parents were related. There were 5 individuals who have a level of inbreeding that is equivalent to if the parents were half first cousins, 10 where the parents could have been as closely related as first cousins, 6 even as close as half siblings, and 1 individual who, I mean, we don't know for sure that the parents were full siblings, but they have a level of inbreeding in their genome that is at that level. So this is actually very high for a wild population, higher than I have seen in any of the other populations of wildlife that I have personally seen before. So why is there so much inbreeding in the flock? Well, clearly, there are a lot of close relatives within the flock. So if we imagine the flock here as all these individuals, and the ones in orange are the ones where we have genomic sequence data, we can take that data and then infer the relationships between individuals and sort of build up the family tree of the flock and figure out, oh, these individuals have a genetic relationship that indicates that they are full siblings. They share both parents. We also have examples of half siblings and cousins, and then a few that just don't appear to be related. But overall, almost all individuals in our data set are related, fairly closely related to 1 or more other individuals in the data set within our 22 birds. So it's like one kind of big family tree where they're all mating with one another, because the flock is isolated and it's not getting a lot of contribution of new individuals. It's just, it was established by a few individuals and has grown over time. But it's not like there are a lot of migrants coming in or new parrots sort of escaping and then joining the flock. That doesn't seem to be that doesn't seem to be occurring on much of a scale. So the other major pattern that I noticed in these parrot genomes was a very distinctive signal of hybridization. And here I'm going to show the genome of Abby. I assume this picture is from when Abby was a youngster, because I don't see any red there. But Abby is one example of the birds that shows regions of very high diversity in their genome, and that is indicative of hybridization between 2 separate species having occurred in this individual's history. So to sort of show this visually, if we imagine here, we have 2 different parrots of different species, one colored gray, one colored black. If they have offspring, then each of their children are 50-50 from each species, right? They might have different, they might inherit different parts of their parents' genomes, but they'll be half one species and half the other species. If one of those individuals then pairs up with a pure representative of one of the species, then the offspring will have not a 50-50 split in their genome, but in this case, they would be like 25% of one species on average and 75% of the other. So we call this a back cross because the hybrid is crossing back with one of the parental species. So you can get different amounts of each species genome and use that to learn more about the hybridization process. And in the San Francisco flock, what we have is they are largely red-masked parakeet, but with some kind of minor contribution of mitered parakeet, which is a separate species. And just to show you, kind of give you some real data here on exactly what the species composition looks like, if I show you, there's this sort of spectrum between 100% red masked parakeet versus 100% mitered parakeet. The wild flock, the individuals that we have sequenced thus far at least, they have 5 to 38% mitered parakeet in their genomes with an average of about 19%. So there are no individuals that are directly a first generation offspring of a mitered and a red masked, but some hybridization like that must have occurred, 2 or so generations previously, and now it's sort of spreading in the flock and creating these like not 50-50, but sort of mostly red masked, partially mitered parakeets. And lastly, I want to come back to, I mentioned that we have these other birds from Sunnyvale and Palo Alto, and also a pet. And it's really great that we were able to include these samples in our sequencing because it gives a lot of context to understanding the San Francisco wild flock. So shown in this picture is one of those, I can't remember if this bird came from Sunnyvale or Palo Alto, but we can see in this two-dimensional plot of genetic similarity that points will cluster based on how genetically similar or different they are, that the Sunnyvale and Palo Alto birds are a very distinct genetic cluster from the San Francisco flock. Like they're completely different. And I don't have a figure to show this, but in my analyses, the Sunnyvale and Palo Alto birds, their genomes are actually at least 60% mitered, and then some other hybrids with some other species or subspecies that we don't yet know exactly what it is. And then secondly, we have this pet individual. And what's interesting about the pet is that we know genetically, this individual is not a hybrid. It is pure red masked parakeet. And unlike the San Francisco flock, it's actually not inbred at all. So it's been, this was one of the surprises for me, was seeing that the Sunnyvale and Palo Alto birds are this completely different hybrid mixture from the San Francisco flock. It's not just like an extension of the San Francisco flock. And although we only have 2 birds at this point, I'm very curious to see if we can get more samples to see if that pattern holds up. So the take-home messages that I want to leave you with are that first, we have this really great high quality sort of reference genome from Acorn and some population level resequencing that has enabled us to reveal a lot of the secrets of the San Francisco wild flock. There are these really interesting and informative patterns in their genetic diversity. The San Francisco flock is a hybrid mixture of mostly red masked and some mitered parakeet, and they're also quite inbred due to high relatedness between individuals. In general, inbreeding is sort of can lead to negative health consequences, but it doesn't always, and that's something that we'll be investigating further in future. But for now, we can say that they are highly inbred. The birds on the southern peninsula in Sunnyvale and Palo Alto look like a completely different hybrid mixture with mitered parakeet and some other as yet unknown species or subspecies. So stay tuned for what we ultimately find with that. We are continuing to analyze the genomic data set to learn more about the Bay Area parrots and sort of reveal these mysteries. We have about a dozen more individuals currently in process for sequencing to expand our data set. And the results from this study, we're getting close to being able to ultimately write up a manuscript and publish it in a scientific journal. And we'll also make those results available on Mickaboo's website. If you want to get in touch with Dara or myself, our email addresses are down there. And I want to give a very hearty special thanks to all of our Mickaboo volunteers and foster carers and donors, especially also especially want to thank Sarah, who's been really helpful with this project and really excited about this project from the beginning. Also want to thank the vets that have helped us procure samples and taking care of the birds, as well as Dara especially, and Ryan and Sherry Chapin at UCSF, our colleagues, and our Stanford University colleagues, Dr. Bernard Kim and Hannah Gellert. So that's my spiel. I'd be happy to answer any questions at this point. Dara, is there anything? I was not paying attention to the chat at all, so I'm sure there were some questions.
Yeah, so there were a few questions that came in the chat, and I think if people would like to raise their hand or add any additional questions, I can relay them to you. I do think that there are some as well that are probably worth bringing up. So one of the questions, and I'm going to work in reverse chronological order as I work up the chat. And so one of the questions says, is it possible that the hybridization of mitered and red-masked parakeets could lead to evolution of a new or a separate flock?
Well, it already has in some ways. Evolution isn't something that has to take thousands or millions of years. I mean, it's happening every generation. So in fact, I would say that in some ways we already have this unique San Francisco flock that has, genetically a genetic mixture of red masked and mitered that doesn't exist anywhere else in the world. And who knows what the future holds for these birds. They've been in the city for a few decades at this point. Clearly they're struggling with a lot of the human impacts around them, like the bromethalin and so forth. But I don't, it's like nature finds a way. So I would say that they kind of already have evolved in some way. Whether they will continue to evolve and persist in the city for millennia into the future, I mean, we'll see. I don't know if that sort of covers it. Dara, if you want to add anything, please jump in.
Yeah, well, another question was, so in the wild, does the size of each species' geographic range prevent inbreeding? Or do you think that there's inbreeding occurring in the wild as well?
It's hard. Well, it's hard to say because we don't currently have really much information about the native populations. Presumably, they are not typically inbreeding in their native ranges, and the population sizes are likely to be at least in the thousands, if not tens or hundreds of thousands, depending. In San Francisco, the flock is only, my understanding is numbers around maybe 250 to 300 individuals, which is a very small population for a wild, self-sustaining species. So that's why there's inbreeding here. I would not expect there to be so much inbreeding kind of naturally in their native range. So yeah, it's because of the isolation and the small population size of the San Francisco flock that there's this inbreeding.
So one individual also pointed out that thus far, it doesn't look like there's evidence of new birds coming into the flock, which, I'd like to highlight that it's great to have rescue organizations like Mickaboo and Animal Care and Control, such that, yes, there are escapees, but there are also, places for these pet birds to go. And keeping in mind that pet birds, while still wild birds, do not have those sort of immediate sort of capabilities. Although, although we don't necessarily know the origins of the flock, do you think at some point we'll be able to sort of understand how many birds founded the flock and potentially when that might have occurred?
Possibly. In some ways, the very high relatedness and inbreeding is an asset for understanding the answers to those questions, but in some ways it can also make things a little bit more complicated. But I would imagine that the number of founders is sort of on the order of maybe a handful of individuals, at least ones that genetically contributed to the flock that we have today. I mean, there could always be individuals that show up and join the flock, but maybe they don't breed, and therefore they're not passing on their genetic material to subsequent generations, or altering the genetic makeup of the flock. And with genetics, all we can really speak to is individuals that are alive in the flock and have genetically contributed.
Well, and Pam has alluded to that I wear two hats. So I'm a researcher at UCSF, but I am also a Mickaboo volunteer. And one of those roles that I have is I actually am fostering some of the I don't want to say former members, because I still feel like they're members of the flock. So Mushi and Julian. Sometimes when these birds come in, their injuries are too, they've sustained too harsh of injuries to be able to survive in the wild. And so they may have varying degrees of abilities to fly or to walk around. And so Mushi and Julian are with me in what I sometimes refer to as home sanctuary, but they are in Mickaboo's foster care along with 400 other foster parrots, give or take, depending on how many requests for surrenders that we get in foster homes. And, you know, I also will say that I had a pet cherry-headed conure for 25 years. And my observations between sort of what it's like to foster a pet versus a wild San Francisco parrot is really kind of night and day, so to speak. I think that there are lots of similarities, obviously, but it really is, you know, there's nature versus nurture. And I think that these wild birds are in fact wild birds. And when we take them into our home, they are familiar with people. They recognize different faces. They will hold a grudge. So if you need to catch them up to take them to a vet, they'll be a little standoffish for a couple of days, right? With you, but maybe they'll like someone else. They certainly, learn. So Mushi and Julian here pictured, they will take treats by hand. They will also step onto a rope perch. So that's actually very helpful for kind of moving around. They play with their toys. There's sort of a lot of similarities. And I think, I would encourage everyone to go out and sort of try to observe them in their natural habitat. And I do think that San Francisco is a natural habitat for them now. It's a new flock. They're living there. They're learning to adapt. They're playing. So a lot of those things that we see in pet birds are shared with the wild flock. But there's also a different way of, I would say, communicating with others or exploring their environment, or realizing that, okay, well, there are birds. So oftentimes if we have a pet, they don't necessarily think they're birds. They think they are humans and part of your flock because they've been raised by humans. But I guess to finish off with the classic example. So and I live up in the Oakland Hills, and so if a hawk comes by, you see Mushi and Julian, they kind of like lay low and a little bit quiet, and they'll start purring a little bit, and the other conures just like stand out and start screaming, and like, they're screaming to each other because there's a hawk out there, and the wild birds are like, okay, no. You're supposed to kind of hunch down and just a little alarm call to each other because you don't want the hawk to see them. So there's some interesting, similarities and differences. And we certainly need more foster homes for some of these birds that are just, unable to live in the wild anymore. All right, so we have another question too. So one of the questions says, so they aren't flying to the peninsula and breeding with those birds, is that correct?
It appears not. With only 2 individuals from the Southern Peninsula, I hesitate to make any strong claims, but just based on what we've seen so far, yeah, it doesn't look like, seems like they're very stay in their little home territories. And I know that the San Francisco flock, they kind of have different spots in the city that they go to, but it doesn't seem like there is genetic exchange between the Southern Peninsula flock or flocks and the San Francisco flock. So they're not flying, the 40 miles or whatever it is down back and forth between San Francisco and Palo Alto. I don't think. That's why I'm really curious if we can get more samples from other parts of the Bay Area and some of those other flocks. What else are we going to find out? Just having these 2 individuals has sort of raised all these other questions that I'm really excited to, dig deeper into.
Yeah. Another question is, are there flocks in other cities and counties, for instance, in the East Bay cities? Maybe I can kind of address that. Again, there's a lot of sort of observations and, my but not anymore. I think that there's also, I think it was Concord, somewhere where there's sort of repeated observations of cherry-headed conures. Now, of course, while we know that it's interesting to think about how, well, even between San Francisco and the peninsula, thus far, only based on 2 individuals, they're not really flying regularly to see each other and potentially interbreed. That maybe these flocks are also, again, like another sort of established start of a colony. I think that there's also a lot of examples in Southern California too. While they have a large number of wild Amazons, there's also a number of red-masked parakeets or miters or some other related species that has also joined them, as well as examples of monk parakeets, which actually living in Chicago, New York City, in Spain. So there do seem to be, these birds are so intelligent and adaptive, and they're trying to find a way in our cities and in our habitats that we have manipulated and changed so much. And kudos to them for being able to survive and make their way in the world that humans have changed so much.
Dara, you cut out, on my end, you cut out for a second. Were you saying there was a flock in Berkeley? I feel like, is that what I've heard before?
Yeah, I do believe that there's been some reports. This is all hearsay. I think that in general, sort of the lack of individuals working in research of these parrots, particularly from their ecological sense and habitat usage. So, we invite people to reach out to us if they have more information about this.
Yeah, it's very helpful to get more. Like, I don't have a personal sort of background with these parrots, but hearing it's really useful to know more from people who have been observing them, and know more about, where they are, what they're doing. And that can actually help our genetic understanding of what we're seeing.
Yeah, I've actually looked around on iNaturalist as well. And so you can put in red mask parakeet, and it will actually map where observations have been. And it's very, consistent with where we sort of think that they are utilizing their habitat. But of course, not a lot of this is, people observing them at 5 a.m. or 6 a.m. at their roosting site, but, you can kind of see the areas of the city where they're using, where they're being observed. And so we're kind of keeping an eye on that as well, to rely on citizen scientists to get more information on where these birds are and how they're doing. So, all right. I don't know if there are any other further questions. We only have a few minutes left. We do have one more question. So would it be worthwhile to recruit volunteers to go out and count these birds in the peninsula and other cities where they might be? Well, as a matter of fact, I do believe that Mickaboo has done that. I think maybe some others have sort of alluded to that. There's sort of the annual bird count, I think, of the flock, done in wintertime.
Is that just in San Francisco?
I think also the southern flocks as well. I would need somebody else to kind of chime in here because I unfortunately am not much of a morning person. So oftentimes I'm not sort of the one to say, yes, I'll go out and count them, but I will look forward to the next one. Again, also in the winter, largely because you see these beautiful green birds with the red heads, and they blend in so well with the foliage. But of course, in wintertime, it's easier to count them because of the lack of leaves. But yeah, so absolutely, we're definitely looking for, if I put my Mickaboo hat back on, looking for more people to volunteer in many ways. So not necessarily just fostering, but also sort of participating in maybe some of these bird counts that, all right, I just about lost my headset. Bird counts, or in other ways, fundraising, because they do also incur a high cost of veterinarian care. So, even if you're sort of interested in more thinking about the wild parrots, we encourage more people to reach out. So, and Linda, I will add your name to participate in a future count as well as myself. All right. Well, and I see that the Pam has put in the chat too that the auction is ending soon. So, Jacqueline, thank you so much for this update, and amazing to see the results of 22 genome sequencing of the red mask parakeet conures, and also showing a great sort of the involvement of Mickaboo with scientific research to support our learning more about these birds. And I will
thank you.
pass it back to Pam then.
Yep, and let me add my thanks to Jacqueline and Dara for their wonderful presentation. A round of applause. Thank you, folks. I'm going to unspotlight you all now, and we'll look forward to future updates as the research continues, of course. All righty, let me get.
Thank you so much for having us.